Hello, my name is Per Lundin. I'm working in NGI production as a bioinformatician, and I'm gonna have this talk about the whole human genome sequencing that we perform in the production unit mainly. So what I'm going to talk about is first I'm just going to go short about the library preparation kits that we have at NGI Stockholm. Karin mentioned those already, so it might be a repeat. And then I'll go through short about the sequencing setup, and then more about the analysis, QC, and delivery of the whole human genome sequencing data that we have. And then I will end with talking about the Sweden project, which was a joint project with us and Uppsala and BILS and so on, just to show what we are capable of doing here in the facilities together. So first about the library preparations and the setups. So in library preparations we have the Illumina TrueSeq, which is a PCR-free protocol. And for that you need 1 microgram of material or DNA to be able to do. If you don't have that much material, you can use the Rubicon protocol, which is a PCR-based protocol, and then you only need 10 nanograms of material. And as Karin said, incoming is the Lucidian NextSeq amplification-free protocol, which is under development from this side. And for that one you need 200 nanograms. And the good thing about this is that it's PCR-free and not as the Rubicon, which does PCR cycles. So the sequencing setup for our whole human genome is that we run it on the Illumina HiSeq X machines on the setup 2 × 150 in read length. Each HiSeq X machine has 2 flow cells that we can run, and each flow cell has 8 lanes per flow cell. So one machine you can run 16 flow cells at a time. And we run 1 sample per lane as the setting now. One run takes approximately 3 days. So our aim when we do the whole genome sequencing is that we want to, of course, maximize the number of reads per lane. And what we have as a limit is that we want to have at least 320 million reads per lane. And that gives us approximately 28.5x or 30x data after duplicate removal. We also aim to have high quality reads. And for the HiSeq X, we need to have at least 75% percent basis over Q30. It's a quality measurement. If it's below that, we can't really use the data. But usually the Q30 is about 85%. We also aim to get low percent of duplicates. Even though we use a PCR-free protocol, we get duplicates, and this is due to the clustering on the flow cell. This is also the number of duplicates is very correlated with the number of reads. So if you have more reads, you most often have more duplicates as well. So this is a trade-off. Also, we aim to have an insert size around 350 bases, because if we have too small insert size, then we will double sequence, as you can see. If you can see the small picture there, then you will sequence double. And if you have too long reads, the quality may get worse, or the clustering may get worse. So why do we aim for 30x, one can wonder? Well, one thing is that, of course, the lanes, or on the flow cell, the lanes, one lane gives us approximately 30x, so it's a nice number for that. But also, when we investigate and look at the number of detected SNPs, germline SNPs, you can see that when you reach about between 25 and 30, that the curve flattens out. So even though we would sequence up to, if you compare 30x or 40x, you can see that doesn't happen that much. So therefore 30x is good enough to detect germline variants. What we also have done is that for the TrueSec PCR-free, we have looked at the complexity of the library to see, and then we used a program called Presect, which predicts and estimates the complexity of the library. And in the best of the worlds, you would like to have the curve following the dashed curve or line there. But as you can see here for the TrueSec, which is the pinkish curve, you can see that it follows it very well and falls off very late. So we have shown that the complexity of our libraries is very good, and that we don't, that we have enough of molecules to sequence. So now I'll come to this is like an overview of the workflow that is done when you do whole human genome sequencing. So the first part here is the blood, that you take blood from a human being in this case. And this is not done by us. And then you extract the DNA from that, and then that is not done by us either. So we come into that we get DNA from the users. We then perform library preparations of this. After library preparations, we sequence it on the sequencing machines, which generates BCL files. The BCL files then are demultiplexed, and we generate the raw data files, as we say, which is the FASTQ files. After that, we run the best practice, which in this case we use an engine called Piper. And that gives us the aligned reach and the variant files. And then after that, we can do a delivery. I will look a bit closer to. So what we use in the pipeline is that we follow the Broad Institute's GATK best practices for germline SNP and indel discovery in whole genome. And I want to emphasize that this is germline. So we are not using the best practices for looking at somatic mutations, like in cancer or in other cases. So the best practice analysis workflow, which is then written in an engine called Piper. So this is an overview. It might look a bit complex for you to look, but if we start at the raw data FASTQ, which is to the left in the slide, so that's the raw data that we get out from the sequencers. So the first thing that the Piper does is that it maps us to the human genome. In this case, we use HG19. After we have the BAM files, we do an indel realignment on the BAM files. After that, we do a duplicate marking using Picard. And the next step is to base quality recalibrate after the realignment using JTK. So now we have processed and aligned the BAM files that one could give to the user if they would like to do more analysis themselves. But we also then perform haplotype, variant calling using haplotype caller. We use it so we generate genomic VCF files. After that, we perform a genotype GVCF and evaluate the genotypes to get a regular VCF file, which we then annotate using SNPs, so we can get the effects of the SNPs. So in the end, what the pipeline generates is BAM files, genomic VCF files, and VCF files. So after this is done, what we do in the bioinformatics group is that we do BP quality control and then create reports to be able to send the data to the users. So for us, quality is very important. So what we always do is that we aggregate all the data that comes from the pipeline. And for that, we use a tool called multi-QC, which is a very handy way of looking at the data from a lot of samples and from a lot of on a lot of different parameters. It's a very quick way. So mainly what we look at is that we look at that the coverage is high enough, that the duplicate levels are not too high. That's the main thing we look at, but then we look at a lot of other things that if they are deviating from the normal, we have to investigate and maybe hold the delivery. We also create a project report which includes all the information about the projects, about protocols used, and so on. So when we deliver the data, today we deliver the data to UpMax, to Meloo, and to what we call the inbox. In the future, however, I don't know exactly when, the delivery will go to Bianca, or actually it will go to something called Grus, but that is not decided exactly when, but it will come in the near future, and everybody will be informed about how to get the data from Grus. Alternatively, we can deliver to a hard drive as well. Due to that, there is a space issue on UpMax or Meloo. We have had to minimize the delivery to the users, meaning that we can't deliver both raw data and analysis data. So therefore, we have two alternatives of delivery. So one is only raw data, then it's only the FASTQ files and the reports. And the other one is that we deliver the BP, best practice analysis. And that's the BAM files, the genomic VCF files, the SNPF annotated VCF files, and also the reports. So now I'm going to jump over to a joint project that we had together, which started up by Ulf Gyllensten, which is called the Sweden project. And we then collaborated with the Uppsala node as well, and also with BILS and Vabi. So the Sweden project was that we sequenced 1,000 individuals. So we sequenced half of them here in Stockholm and half of them in Uppsala. Then we performed the regular data analysis, mapping and variant calling, as I just mentioned to you. But after that, we did an extra step where we did a joint calling on all the individuals together. And from this, we could get genotype frequencies that then we would like can be available for download for the community. It can be downloaded from the link as you see below. Just short about what was found from the Sweden data or in the project. So we could found a total 33 million genetic variants, and where the majority was SNPs, and just 4 million of them were indels. Out of these 33 million, almost 10 millions were novel, meaning that they have not been reported previously. And in this case, we compared them to DBSNP 147. Out of these ones, interesting, 26,600 approximately of the novel variants are a change in protein sequence, which could be very interesting in the future to look into these ones. And if you look on individual basis, around 7,200 novel variants were detected in each individual. So the Sweden project, the aim of this project was to identify a cohort that reflects the genetic structure of the Swedish population, and then generate WGS data using short and long read MPS technologies. and make the information available to the research community and the clinical genetic laboratories through a user-friendly database. And I would say all of these aims have been fulfilled. And so this is just to show that this was a collaboration with the National Genomic Infrastructure here in Stockholm and in Uppsala, which did the sequencing and data analysis, and then NBIS, that did the data analysis and the distribution. And then also the Swedish Twin Register, which provided samples for sequencing. So that was all I had to say. Thank you.